listening. If you're already in, love to hear where you're coming in from. I love to travel, so I've lived vicariously through the countries I see on the screen. So I'm coming in from Nashville. Looks like Feliz is, if I'm pronouncing your name correctly, coming from Florida. Hey, you have some fellow Californians out there, Mike. So, okay, wow. Belgium. Are you? I don't know if you're seeing the chat coming in. We even have some Saudi Arabia. Very cool. We'll give it just another minute just to make sure everybody has a chance to come in. And another thought might be, if is this your first Greenlight Guru event? And if it's not, I'd love to know what your favorite topic has been so far. Love to see something like that if you have any feedback. First event, Beatrice. Well, welcome. All right. All right, it's 11:01, so let's go ahead and get started. My name is Etienne Nichols. I'm a medical device guru here at Greenlight Guru. I'll be the moderator for today's event. Today's webinar is planned to run for about 90 minutes in total. We definitely want to hear from you. Looks like most of you have already found the chat tab, which is great. Keep those answers coming in, whether it's, I'd love to see where you're from, but also love to see what your favorite topic is and maybe some topics you'd like to see in the future. Let us know. Helps us make sure that the content we're giving you is relevant to what your needs are. But just a quick overview of what those tabs are. So the chat, feel free to talk amongst yourselves, give us some feedback, but the Q&A tab is where you can ask specific questions that you want Mike to answer during the Q&A portion of today's webinar. One question we always get is, will you be sharing the recording and the slides after the webinar? Yes, we will be sharing that recording and the slides, so keep an eye out for that email from us. That should be coming later today or later this week. I'd like to share a few words about Greenlight Guru and why we put on these free training sessions. Improving the quality of life for our customers and their patients is our mission here at Greenlight Guru. It's likely a similar mission to many of you joining us today. We're constantly looking for ways to fulfill that mission, whether that's through hosting these free webinars, partnering with world-class medical device experts like today's presenter, Mike, or through our award-winning cloud-based solutions. At Greenlight Guru, our focus is on moving medtech forward. Our solutions are designed to help streamline your processes so you can navigate the regulatory hurdles and succeed in the market. Our MedTech suite provides quality management, product development, and clinical data management solutions, along with high-quality training and education designed exclusively for medical device experts. So if you're interested in learning why over 1,000 of the world's leading MedTech companies trust Greenlight Guru, feel free to head over to www.greenlight.guru after the presentation and check out a personalized demo. All right, let me give you an introduction to your presenter today. Sounds like some of you, this is your first time, so maybe you're not familiar, but many of you will likely, this is likely a familiar voice. Dr. Mike Drews is president of Vascular Sciences, a consulting and training company offering a broad range of services to medical devices, pharmaceutical, and biotechnology companies, including creative regulatory strategy and competitive regulatory intelligence, regulatory submission design, FDA presentation, preparation, and defense. Dr. Drews is an internationally recognized expert who brings a breadth of knowledge and hands-on experience in his teachings from the ongoing work that he does with the FDA, Health Canada, and other regulatory and governmental agencies, and around the world. He's also a featured keynote speaker on cutting-edge medical technologies and regulatory affairs. And if any of you are familiar with the Global Medical Device Podcast, you'll recognize his voice right away, as he's a longtime, frequent guest on the show. And with all of that, I'll let you take it away, Mike.
Thank you, Etienne, for that very kind introduction. And I just want to confirm that you're still able to see my screen. Is that correct?
Yes, definitely.
Okay, terrific. Thank you. All right, so welcome everybody to this webinar on significant risk versus non-significant risk devices. What's the difference and why does it matter? As Etienne just said, my name is Dr. Michael Drews. You can call me Mike. The only thing that I want to reiterate about my background that Etienne mentioned is in addition to consulting for a wide variety of medical technology companies, I also work as a consultant for the FDA as well as Health Canada and a few of the other regulatory agencies in the world. And the reason why I mentioned that is, as some of you may appreciate, that's not something that a lot of people could say, that work on both sides of that proverbial fence. And so I really try to use that to my advantage. And in addition to my consulting and my teaching, as Etienne just mentioned as well, I'm a contributing editor to several of the largest medical technology and regulatory publications in the world, including a very active relationship with Greenlight Guru. Etienne mentioned the podcast series that I do, and here's some other resources that you might want to take a look at as well if you're interested. I've done podcasts and webinars and columns on a very, very wide variety of topics. And then the last thing that I'll just mention, this is not bragging, but just as a FYI, I'm kind of proud of this. In December of last year, I was named one of the top 24 MedTech voices to follow in the current year, 2024, by QMed, the publishers of MDDI magazine, as well as some other publications as well. So that's a tiny bit about me. Let's, before we get into the subject matter, I just want to see kind of where we are as an audience. So I have a couple of polling questions, and the polling questions that you have, if you can make them visible, Etienne, are a little bit different than what you see on the screen right now. So Etienne, why don't you announce the first question, please?
All right, just open that poll. Hopefully they're able to see it.
I think the essence of that first question is, are you working on a significant risk device or a non-significant device, or you don't know? Non-significant risk device.
Yeah, we have the votes coming in. We'll let it stay open for a few minutes, but it looks like it's about 50-50 split between significant risk and non-significant risk, with about a quarter of the I don't knows. So
okay. And
yeah.
by the way, for those of you that did answer I don't know, I really appreciate your honesty and your candor there. So admitting what you don't know is not something that a lot of people feel comfortable doing. Shall we move on to the next question, Etienne?
Sure thing. Let's close that one out. Opening the last one. Okay, it should be open. Let's see if we get any answers here. Okay. Do you know the difference? 20 votes so far as no, and 8 votes yes. Okay, it's and it's climbing. So we're about 60-40. The majority no.
Okay, well, once again, kudos to those of you who said that you don't know the difference, and that's the purpose of today's webinar, or at least one of the purposes. And by the way, one other thing that I should mention quickly about my background before we continue on and close the poll is, because we're talking about risk in this webinar, just wanted to mention that I'm a subject matter expert for FDA in a few different areas, one of them being risk. And so this is an area that's near and dear to my heart, pardon the pun. All right, final results on the poll, Etienne, before we move on?
59 votes yes, 74 votes no, so about 60-40 still, leaning heavily towards no.
Great. Thank you, everybody who participated. That gives me sort of a sense of where we are as an audience. So very quickly, in the limited time that we have together today, I want to try to talk about as many of the following things as possible. First of all, what is a significant risk, or SR, and a non-significant risk device? How does the SR versus NSR system compare to other systems of risk that we have here in the United States? As we'll talk about in just a moment, we actually have 3 different independent but also interdependent systems of risk here in the US. Whether we need 3 or not, that's a different discussion, but we have 3. Who determines if my device is significant or non-significant risk, and what is that determination based on? Why does significant risk and non-significant risk matter? What are the requirements of each? If my device is SR, what do I do? Alternatively, if it's not, if it is NSR, what do I do? How and when should we take my SR versus NSR determination to the IRB? That's the Institutional Review Board, or boards plural, and or the FDA. Does my device need a clinical trial? And that's a topic of a whole other webinar, but I'll touch on that a little bit. How should I select clinical trial sites and IRBs? Once again, that's a topic of a whole other webinar, but I'll touch on that just a little bit. Should I do a pre-submission meeting or a pre-sub with FDA for an NSR device, even though it's not required? And then we'll wrap up with talking a little bit about the challenges of the future. Threw in a bonus question, is there a template for making this determination? And I'll be throwing in a litany of tips and tricks and as many things as I possibly can in the time that we have together. And then we'll wrap up with some final thoughts and some Q&A at the end, and then we'll send you on your merry way. So we have a pretty ambitious agenda in the time that we have, so let's get right into it. So the first question is, what is a significant risk or non-significant risk device? How do they differ? And I'm going to show you what FDA thinks, that is what's in the CFR and what's in the guidance. But at the end of the day, what is most important is what you think. Do you agree with what's in the CFR? Do you agree with what's in the guidance? Do you agree with FDA? So starting out with a significant risk device. What is a significant risk or SR device? Under the regulation, there are four requirements or parameters, if you will, that make the SR determination. First of all, is the device intended as an implant and it presents a potential for serious risk to the health, safety, or welfare of a subject? So what's an implant? And notice specifically, they do not say permanent implant. You know, one of the things that I've learned in 30 years of playing this game is don't just read what the regulation says, but also read what the regulation doesn't say. Because oftentimes what the regulation does not say is just as important, sometimes even more important than what it does say. So it says it's intended as an implant. For those of you that are sort of regulatory geeks, what is a permanent implant? Well, a permanent implant is not what you think it is. A permanent implant is not necessarily something that is intended to last you the rest of your life. And a permanent implant, in regulatory terms, at least here in the United States, is the indication is greater than 29 days. That's it, nothing more. Greater than 29 days or greater than a month. Breast implants, for example, were never intended to be in the patient's body for the rest of their life. So for a SR device, it's intended as an implant, but it doesn't say what kind of implant. The next requirement is and I don't know why they changed the verbiage here, so I put in, instead of purported or represented, in regulatory terms, what we mean there is intended to be used for supporting or sustaining life. And by the way, the sections in green there are just copy and paste, so I'm not going to read those each time. But what does supporting or sustaining human life mean? For those of you that have heard any of my previous webinars or podcasts, you may remember that I've said many times that regulation is about two things. One is about the interpretation of words, and part two is your ability to defend your interpretation. So let me say that again. Regulation is about two things. The interpretation of words, that's part one. Part two is your ability to defend your interpretation. Here are the words on the screen. And by the way, this is not guidance. This is the CFR. This is the Code of Federal Regulations. And by the way, this has not changed since at least 2006. So this has been around for a long time. So you can interpret these words, in this case, supporting or sustaining human life, in many, many different ways. But then once you come up with your appropriate determination, of course, you've got to go to the FDA, and in some cases, you've got to go to the IRB and sell your interpretation to them. The third requirement is the device for use or intended for substantial importance in diagnosing, curing, mitigating, or treating a disease or otherwise preventing impairment of human health? Well, what the heck does substantial importance mean? I mean, can anybody name a company or a device that they're spending time and money to develop that they would not argue is of substantial importance, at least to somebody? And further, what does impairment of human health mean? I know I'm kind of going quickly and I'm offering these as sort of rhetorical questions, but I'm simply trying to illustrate how this idea of regulation is about two things: the interpretation of words and your ability to defend your interpretation applies to this particular subject matter in this webinar. And the last requirement, which I think is total redundancy, is it simply, reiterates the tail end of the previous three requirements. But one comment to make here is, why limit ourselves to the subject or the patient? From a risk perspective, sometimes, actually oftentimes, not only do we need to consider risk to the patient, but we also have to consider risk to the caregiver, to the physician, to the nurse, to whoever it is that's using the device on that particular patient. According to the strict interpretation of the regulation on the screen right now, the letter of the law, so to speak, that is not taken into account in the significant risk versus non-significant risk determination. However, in my opinion, according to the spirit of the law, it's definitely in there. So there's a difference between the letter of the law, that is what the regulation says explicitly, and the spirit of the law, or what I like to call the regulatory logic. Examples of significant risk devices include things like certain surgical lasers, not all, tissue adhesives, cardiac bypass devices, intravascular stents, and so on. There's a long list. Incidentally, the guidance for this topic, which I'll show you later in the webinar, it's a pretty short guidance, only about 15 pages long, about 4 or 5 pages. The last 4 or 5 pages of the guidance is no more than a laundry list of examples of significant and non-significant risk devices. But one thing to note, and I see a lot of people make this assumption, is that a significant risk device is not necessarily a class 3 device. We'll talk about the different systems of risk that I mentioned in just a moment. But a lot of people think that a significant risk device must be a class 3 device. In fact, there are a lot of class 2 devices that we call moderate risk devices that are that's in the classification, but in the SR versus NSR system, we call them a significant risk device. So how can something be moderate risk in the classification system and significant risk in this system? Well, I don't know. Quite frankly, it's sort of a schizophrenic argument, but the easiest way that I can explain it is that the systems are different. Risk does not mean the same thing in the classification system as it does in the SR versus NSR system, as it does, as we'll see, in the medical device software system. But again, we'll get to that in just a moment. Shifting over to the other side of the audience that is working on, or they were working on, non-significant risk or NSR devices, according to the regulation, and again, this is not guidance, but according to the regulation, I wish I could say that I'm joking here, I'm not. The definition, the criteria of a non-significant risk device is that it, quote, does not meet the definition for a significant risk device, end quote. Nothing else is said about what an NSR device is, other than, of course, giving a laundry list of examples. So my question to you all in this audience, and this is sort of a rhetorical question, does this help? Unfortunately, we frequently will define something in the regulatory world, and we don't focus on what that thing is. Instead, we focus on what it's not. So I don't know what a non-significant risk device is. All I know that it's not is that it's not a significant risk device. I don't know how you all feel, but this is a pretty bad way to define anything. It's kind of like, I don't know what an apple is, but all I know is that an apple is not an orange. Right? That's a pretty weak way to define it. So my advice here, and I'll get to this in a second. So examples of NSR devices include contact lens solutions. Now, again, I need to just make clear here. These are examples that I just pulled from FDA's guidance. I do not necessarily agree with the examples here, but these are the examples that some of the examples that FDA lists in their guidance. So contact lens solutions, these are, liquids that you put into the eye. Should that be an NSR device? Well, there's a lot of problems, or I shouldn't say a lot, but there are problems with contact lens solutions, and that can cause problems to the eyes and everything else. Dental filling materials. These are permanent implant. One could easily argue that no permanent implant should be a non-significant risk device, because what is going to happen to that permanent implant after it's been in the patient a year, 5 years, 10 years, maybe 25 years or even longer? Nobody, no matter how much testing you do, nobody can make that determination, at least in advance. Digital mammography, Again, one could argue that that's not necessarily a NSR device, because if it conveys the wrong information to clinician, they might say that the patient does not have breast cancer when in fact they do. That would be a false negative. Or similarly, they might conclude that the patient has breast cancer when in fact they don't. That would be a false positive. So there are risks there, but they're not taken into account in the SR versus NSR system. And urethral stents. You may remember a moment ago when we talked about significant risk devices, an intravascular stent, a stent that we put in, say, a coronary artery or a carotid artery or iliac artery in your leg. Those are significant risk devices. However, a ureteral stent, a stent that we put in either the urethra or in the ureters, is a non-significant risk device. So depending on where the in the body the stent goes and what it's doing, that may determine whether it's significant risk or non-significant risk. But here's the most important part of a message on this slide. Since there is no specific definition for an NSR device, it just says that it's an NSR device doesn't meet the definition of a SR device. My best recommendation, and some people like this recommendation and some people don't, always, always, always start by assuming your device is significant risk. Even if you think it's a non-significant risk device, assume that it is a significant risk and then work to prove that it's not, that it doesn't meet those 4 criteria that I just went through with you a couple of minutes ago from the CFR. For those of you that remember your high school geometry class, it's the regulatory logic of an indirect proof. In other words, we prove what it's not, and then whatever's left is what it is. That's the logic that I'm using here. I'm proving that it's not SR, it's not significant risk. Therefore, once I prove it's not significant risk, then it must be, sorry, let me say it again. Once I prove that it's not significant risk, then what remains is it must be NSR, non-significant risk. I know that's a little bit confusing, but I hope most of you understood what I mean. Start by assuming that your device is SR and then prove that it's not. And by the way, this is exactly what the IRBs, as we'll be talking about in a moment, should be assuming as well. Every single device that comes to an IRB, they should, if they're knowledgeable and if they're doing their jobs, they should assume that it's significant risk. And then it's the burden of proof is on the company to convince the IRB that it's not significant risk. That's the way that this game, in my opinion, should be played. How does the significant risk and non-significant risk systems compare to other systems of risk? Well, suffice it to say, they do not compare at all, other than simply by giving us 3 different methods or 3 different systems, if you will, of categorizing risk. And so just to explain what I mean, I did a podcast for Greenlight a while ago on specifically the 3 systems of risk here in the United States. And if you're not familiar with that podcast, there's a link in the handout, or just go to Greenlight's webpage. You can listen to that. But here are just some quick highlights from that particular podcast to help differentiate between these different systems of risk. So as I said a moment ago, we have 3 systems for risk. The classification system, which is probably the one that most of you are familiar with, class 1, class 2, class 3. The SR versus NSR system, that's obviously the topic of today's webinar. And finally, we have the basic or enhanced documentation system for software. This was formerly known as the software level of concern system, that some of you may remember it from a year or so ago. On the classification side, and by the way, just as an FYI, I've done many webinars for Greenlight over the years. This is one of the most listened to, understanding the medical device classification system from basics to beyond, using the classification system to your advantage. So if you're not that familiar with the classification system, or even if you think you are, you might want to consider watching that webinar. If you can't find it, just send me an email or contact Greenlight. I'm sure that we can point you in the right direction. So the classification system is just one way of stratifying the layers of risk. In other words, we break them into layers, or what we call classes. For example, class 1, class 2, class 3. As a general rule, the higher the class, the higher the risk. The lower the class, the lower the risk. So as a general rule, class 3 devices are of greater risk than class 1 devices. But the problem with that is, although that's what every textbook says, and that's what virtually every regulatory consultant says, there's two problems with it. First of all, it's a gross oversimplification. It depends on what kind of risk you're talking about, and there are a ton of exceptions. A ton of exceptions. Average regulatory professionals know the rules. The best ones know the exceptions. And there are a ton of exceptions. I don't have time to talk about exceptions in this webinar, but I've talked about many exceptions in my other webinars and my podcast, so I would refer you to those other sources. Classification in the US does not necessarily translate linearly to classification in the EU or elsewhere. In other words, if you have a device that's, say, class 2 here in the United States, it may or may not be class 2 in the EU or Canada or somewhere. They're similar systems, but they are philosophically different. In other words, they are not substantially equivalent, pun intended. Once again, I don't have time to go into the differences in the classification systems. That's beyond the scope of this particular webinar, but it's really unfortunate that we use this similar terminology, class 1, class 2, class 3, in the EU, class 4, and so on, in different parts of the world, because it really confuses people, and they're really not the same. Of course, the topic of today's webinar, which we're going to be digging into more and more as we continue, is the SR versus NSR system. As we'll discuss, this is very important in clinical trials because it will help determine if your device is going to require what we call an investigational device exemption or an IDE, or is it what we call IDE exempt? And the determination, might be a bit of a surprise to some of you, determination of the significant versus non-significant risk status is not made by the FDA. It is not made by the IRB, or the IRB has to agree with you, but the determination is not made by them. It's made by you. It's made by the medical device company. You have to determine yourself whether your device is SR or NSR, and then you need to go to the IRB, and in some cases you need to go to the FDA and sell it to them. Is this the proverbial putting the fox in charge of the hen house? Absolutely, of course it is. But that's exactly what the regulation says. And then finally, the third system of handling risk is the what was formerly called the software level of concern system, which had class A, B, and C. That was recently updated to the basic enhanced documentation system. Basically, the only reason why that's important pragmatically is because it will determine the level of documentation required for your software. So that's kind of the idea of that. Do we need 3 different systems for risk here in the United States? Well, remember, I'm a subject matter expert for risk for the FDA on risk. So this is a topic near and dear to my heart. On one hand, risk is an incredibly complicated, incredibly difficult subject. A lot of people that think they understand it, but they really don't. But on the other hand, it seems like we go out of our way to make it even more complicated than it needs to be, for example, by creating these 3 different systems of risk. Just a couple of general reminders about risk. The default classification for any new medical device is class 3. So the example I like to use is a Band-Aid. If you're coming to the FDA with a Band-Aid for the very first time, it would be a class 3 device. We would be treating it essentially like other class 3 devices, like artificial hearts. And that makes no sense to anybody that has an IQ of more than 5. So that's when we would use probably a de novo, because a de novo is a form of down classification. And I've done lots of webinars and podcasts on the de novo. And then the other reminder from that podcast I wanted to share with you is that when it comes to the labeling, you can say anything that you want. You can claim anything that you want. If you want to claim, and I'm going to use this example, as an extreme, but if you want to claim that your device cures cancer and regrows missing limbs, fine. I have absolutely no problem with that. But before you do, convince me, show me your data, convince me as a biomedical engineer from an engineering and a biology perspective that you can prove it, that you can support it. You can say anything that you want as long as you can support it. And then the other thing to keep in mind, especially here, is if we have the same technology but different labeling, different indications, risk may also change. And your NSR versus SR determination may also change. It is possible to have a device for one indication that is non-significant risk and exactly the same device for another indication that is significant risk. And when I say exactly the same device, I literally mean exactly the same. In other words, what the device does, how it works, its mechanism of action, exactly the same. It's just what it's intended that's different. Bottom line, although we do have 3 systems of risk here in the United States, please keep in mind they are all independent of one another, but they are also interdependent on one another. And unfortunately, there's no way, certainly no easy way, to convert from one to another. There's a conversion factor or equation. If you want to convert, say, from Fahrenheit to Celsius, there's a little equation that we can use to do that. There is no simple way to convert between the classification system versus the SR and NSR system versus the base documentation system. Believe me, in some ways I wish there was, but there's no easy way to do that. For those of you that are more interested in risk, I would point you to some of my many resources on risk, the previous podcast that I just mentioned. And here's a webinar that I did a few years ago, but it's one of the most popular ones, many connotations of risk and the consequences of getting them wrong. This is where I introduce and I describe in some detail my 3-bucket approach to dealing with risk, because as a subject matter expert for risk for FDA, I think that all of the systems that we use for risk in our industry, including the ISO system, is woefully inadequate. Even though these are the industry standards, they are woefully inadequate. Many years ago, I developed my system. I call it this 3-bucket system, and it is now being recognized by the FDA in a growing number of submissions. So if you're not familiar with that, you might want to check that out. Continuing on, and by the way, as a reminder, hopefully all of you are thinking about questions to ask at the end of the webinar, so feel free to type those into the chat and we'll get those, or as many as we can, in the Q&A section at the end of the webinar. So continuing on to the next question, who determines if my device is SR or NSR, and how is determination made? Well, as I hinted at a few moments ago, it's not the FDA that makes this determination. It's not even the IRB that makes this determination, even though in the regulation it kind of says that the IRB makes the determination, but they really don't. I'll explain that more in a moment. It's you. It's the medical device manufacturer. It's all up to you. Once again, is that the proverbial putting the fox in charge of the henhouse? Absolutely. I don't think anybody can argue that it is, but that's exactly what the regulation says. So why does the SR versus NSR determination matter, and what are the requirements of each? Well, for significant risk devices, we need to submit an IDE, an investigational device exemption, to the FDA, and that IDE be approved before we begin the clinical trial, at least here in the United States. In other places in the world, that's different. We're talking about the US. So that's requirement number 1. SR device, you need to submit an IDE and obtain approval of that IDE before beginning your clinical trial in the United States. It's beyond this particular webinar to talk about what goes into an IDE. Maybe I'll do another webinar on that in the future. But for now, I did provide to you at the bottom of the screen a link that will take you to FDA's IDE approval process, and it will tell you there, some of the components that go into the IDE. In addition to having an approved IDE, before beginning your clinical trial for your SR device, you need to submit your investigation plan, meaning your protocol and everything else, to the IRB, and specifically to each IRB. So for those of you that are not familiar with IRBs, or institutional review, there's a quick primer on that. And by the way, I just did a podcast on this for Greenlight very recently. I checked and it has not been published, I believe, yet. So hopefully within the next couple of weeks, There will be a podcast getting into the IRB and clinical trial process in much more detail. But in a nutshell, an institutional review board, or unlike the FDA, where FDA's authority is nationally across the entire country, the IRB's authority is local at that particular institution, hospital, medical school, whatever it is. And the other thing to remember about the IRBs is that the decision of each IRB is independent of each other IRB, and it's also independent of the FDA. So it's possible, for example, that the IRB and the FDA come to different conclusions as to whether or not your device is significant or non-significant risk. It doesn't happen often, but it does happen. So, for example, if you're going to do a clinical trial, let's pick a number. Let's say 3 clinical trial sites. And FDA often likes to see clinical trials at 3 different sites. Don't ask me why. That's a different discussion, but they like to see 3. In order to begin your clinical trial for an SR device at 3 different sites, you actually need 4 different approvals, if you will, or blessings, as I like to call it, because strictly speaking, the IRBs are not approving your study. You need to get the IDE approved. That's number 1. And then you need to get each of the IRBs at those local institutions. So, picking some names at random, let's say, you're going to do your clinical trial at Hopkins, at Stanford, and at Harvard. Okay, I'm just picking those 3 because they're pretty well. You would need to get the blessing of the IRB at each of those 3 institutions, unless, as I said before, average regulatory professionals know the rules. The best ones know the exceptions. What I just said is true if you're dealing with a traditional IRB. If you have an option for what we call a commercial IRB, an IRB for hire, so to speak, and if all 3 of those institutions, Harvard, Hopkins, and Stanford, will all accept the same commercial IRB, it's totally up to them whether they will or not. But if they accept it, you don't need to deal with 3 different IRBs. You can only deal with 1 IRB. So you can get your number of approvals down from 4 down to 2. If they all accept the commercial IRB, and as I think I say in one of my upcoming slides, make sure to find out if the institutions that you're looking at doing your trial at recognize any of the commercial IRBs. If, on the other hand, you're working on a non-significant risk device, you still need to get the approval of each of the IRBs. You always have to get the approval of the IRB, but you do not need to get the approval of the FDA. In other words, you do not need to get an IDE. If your device is NSR, it's what we call IDE exempt. But my advice, usually, I would say, greater than 95% of the time, if a company is working on an NSR device, my advice to them is to take it to the FDA anyway, even though it is absolutely not required. Let me say that one more time. Most of the time, and I work on a lot of NSR devices, most of the time when I am working with a company on an NSR device, I will recommend to them take it to the FDA anyway in advance of the clinical trial, in the form of a pre-submission meeting or a pre-sub, even though it's not required. Why? Because, as we're going to see in an upcoming slide, but just to do a little foreshadowing here, I want to minimize, if not eliminate, 2 very common problems I see companies run into. The first problem is a sanity check. I want to make sure that because the NSR determination is up to me, I want to make sure that FDA it that way. In other words, it is possible, and I've seen this happen before, that a company determines their device is NSR. They take it to their IRBs. The IRB says that it's NSR. Then they submit it to the FDA, and the FDA says, gee, I don't know what you all are smoking. You must be smoking your socks. We don't see this as an NSR device. And now you have a bunch of problems before you even begin talking about your device. This is a very easy problem to mitigate, if not eliminate it, by taking it to the FDA first as a pre-sub. The second and even more common reason why I like to take NSR devices to the FDA, even though I'm not required, is to eliminate a very, very common problem where a company does a certain number of tests, measure, for example, a certain number of endpoints in their clinical trial. They submit their trial results to the FDA. The FDA comes back and says, gee, we want to see these additional 1 or 2 endpoints. And now you have a whole clinical trial over again to collect those additional 1 or 2 endpoints. Can you say ka-ching, ka-ching, ka-ching? This is such amateur, such an elementary school level mistake. But unfortunately, I see it happen in some cases to the largest medical device companies on earth. I'm thinking to myself, you've got, dozens, maybe hundreds of regulatory professionals running around in your company. What the heck are they getting paid for if they make that such a basic mistake? We'll talk about this a little bit more later in the webinar. Just a couple of reminders. The regulatory logic, and I'm not exactly sure I agree with this or not, but the regulatory logic on the NSR is FDA considers an NSR to have an approved IDE. And please notice I'm putting that phrase approved IDE because it is not approved IDE. When the IRB agrees with the NSR determination and approves the study, which begs the question, how do you show this? Recently, I had a company in a situation where the IRB determined that or agreed with the company that the device was NSR, but the investors, this was a small company with VCs, they wanted some proof, a document from the FDA to saying that this is not NSR. And I said, well, sorry, that this is NSR. And I said, no document that exists. There's no mechanism, at least that I'm aware of. If somebody knows of one, please share it with me. But I'm pretty sure there's no mechanism to go to the FDA and get some determination that your device is in fact NSR. There should be, and I've made that suggestion to the FDA many times in the past. And if any of my FDA friends are listening to this webinar, and I know that you do, even though you admit to that publicly, maybe you'll take that suggestion back because I've made it many, many times. So in that particular case, the way you show it is by your meeting notes and by your pre-sub meeting notes. In other words, you mention in your meeting notes that it was discussed or it was presented as NSR and FDA or the IRB agreed that it was NSR. That's about the strongest proof. It's not real strong proof, to be honest with you. I wish there was a better way, something along the lines of a 510K clearance letter, something like that. But we don't have that, at least not yet. Finally, if the IRB disagrees with you, in other words, if you go to the IRB and try to convince them that it's NSR and they disagree with you, according to the regulation, if they formally let you know that we don't think this is NSR, then you, the manufacturer, need to notify the FDA within 5 working days of that notification. This is exactly what the regulation says. However, this is strongly to be avoided. Strongly to be avoided. Why? And I have to be a little careful how I explain this, if the IRB issues you a letter or a document that says, we disagree that this is NSR, or we're not sure if it's NSR, then under the regulation, you have that obligation to notify FDA within 5 days. It's, there's no ifs, ands, or buts about it. However, and this is part of the IRB manufacturing, if it looks like it might be going in that direction, if you get a feel for whatever reasons, and once again, I have to be really careful what I say here, that the IRB might not be convinced that it's NSR device, then what I do in those situations, and this doesn't happen very often, but what I do is I withdraw the IRB submission from that institution before they give me a formal determination. Why? Because then that allows me to do what we affectionately call IRB shopping. In other words, I can say to my friends at Harvard, okay, Harvard, we would really like to work with you, but if you're not, on board with this NSR determination, quite frankly, it's not worth it to work with you. We're going to go down the road to Boston University or Tufts or somewhere else. This is what we call IRB shopping. And it happens fairly frequently. It's not a foreign concept, by the way. For those of you that do business in the EU, it's the same thing with notified bodies and notified body shopping. The regulation says we have to use a notified body, but the regulation doesn't say which one. So we can shop around and choose the one that And even here in the United States, there's regulatory precedent for this. For those of you that are familiar with the third-party review program for the 510K, it's exactly the same thing. If you have certain kinds of low risk that are eligible for what we call third-party review, and I don't have time to get into the details of that here, then we can look for a third-party reviewer, shop for a third-party that we want to work with. It says that we have to work with one. It doesn't say which one we want to work with. So although this might sound surprising, but to some of you, this concept of IRB shopping, it's absolutely not without precedent. I just gave you two examples. One is notified body shopping, and the second is third-party review for 510K shopping. Next question. If device is SR, what do I do? Or alternatively, if it's NSR, what do I do? So just to recap, because I know this is a little confusing to some of you, if your device is SR, you notify the FDA via an IDE, and you also need to notify all of the necessary IRBs. If, on the other hand, your device is NSR, you're not required to FDA. In other words, you're IDE exempt, although, as I said a moment ago, in most cases, I still strongly recommend to my customers to notify them anyway for reasons that we'll talk about further in a couple of minutes. But you still have to notify all of your IRBs. So at the end of the day, you always have to notify the IRBs. That goes without saying. The only question is, are you required to notify the FDA or not? And if so, are you IDE? Are you required to submit an IDE, or are you IDE exempt? So when and how should you take your SR versus NSR determination to the IRBs and to the FDA? Well, my answer is, my advice is sort of schizophrenic, but it's exactly the same as what I say when people ask me the same question about meetings or precepts. How early in the process should I go? On one hand, I say it's never early to go to the FDA or to the IRB to begin this process. But on the other hand, I say don't go, and I'm sorry, there's a typo in that slide. Don't go anywhere, anytime earlier than when you are fully prepared, not just to present your plan, but to vehemently defend it. The way FDA is supposed to work is, I don't know where what the weather is where you are, but here in San Diego, it's almost always, every day is almost always blue sky and the sun is shining. So if I go into the FDA and I say the sky is blue, FDA's job is to say, okay, prove it. That's the way that this game is supposed to be played. Notice that FDA is not saying we don't think the sky is blue. That's not the same thing. FDA is simply saying, oh, you're saying the sky is blue. You need to prove to us that the sky is blue. In other words, it's the old Ronald Reagan mantra, trust but verify. The job of a good regulatory professional in a company. Make sure that the company can, you can trust but verify whatever it is that you're saying, or in this case, what you're doing to the IRB or to the FDA. At the very least, you need to begin as early as possible to start to identify your clinical trial site or sites, usually 3, sometimes more, sometimes less, but not very often. You need to identify if you have the option for using a commercial IRB, and there are advantages and disadvantages to using traditional versus commercial IRBs. You can listen to that podcast that's going to be published shortly, where I go into that in much more detail and so on. In other words, you need to get all of your ducks in a row. There are a lot of logistical ducks, including but not limited to the SR versus NSR determination that you need to get in order to be able to begin your clinical trial. If you want to be a little more specific, here's an equation for you. Take the anticipated start date of your trial at the particular institution that you're dealing with. Subtract from that date the average IRB approval time at that particular institution. Don't take some generic IRB approval time, but you can ask the clinical institutions, how long on average does it typically take for IRBs to make a decision, an approval or a not approval? They should be able to easily give you that information. And further, here's another nuance you should be aware of regarding If you're dealing with a large institution like Stanford, for example, Stanford has multiple IRBs. They might have one IRB for cardiovascular products, second one for neurological products, a third one for oncological products, and so on. So if you're working on a cardiovascular device, an intravascular stent, for example, make sure you ask them, what is the average IRB approval time for the cardiovascular IRB at Stanford? You don't want to be commingling those numbers with neurology IRB and the gastroenterology IRB, because that's going to really skew your analysis. And if they can't give you that information, that would cause me to hesitate as to whether or not I really want to work with them. So take your average IRB approval time, multiply that by a safety factor. As always, as engineers, when we design anything, we always have to build in there a little bit of a safety factor. The safety factor you use, obviously, that's up to you. I would say maybe, to be safe, something like 50%. So if they tell you, for example, the average approval time for a cardiovascular IRV at Stanford is, let's say, just going to pick a number, 75 days, just to be safe, you might want to, increase that by 50%. Just give yourself, some leeway. So there's a equation that you can use there if you choose to do so. Next question, and we're coming up, we'll be wrapping this up fairly soon, because I want to keep the last 15 minutes or so for Q&A and discussion. Does my device need a clinical trial? Well, this is a fascinating question to me, and I get this question almost every single week, almost every single day, in fact. Many people think this is a decision. It is absolutely not a regulatory decision, at least not completely. The main decision is from a biology and an engineering perspective. And please don't tell me that your working device, the previous devices did not need a clinical trial, therefore I don't need to do a clinical trial. That is a very poor excuse for not doing a clinical trial. And if I was sitting on the A side of the table, I would be all over you like a cheap suit. Alternatively, I see a lot of people make the opposite assumption, that the similar devices have gone a clinical trial, therefore I must undergo a clinical trial. Nowhere in the regulation is the regulation that explicit, and in my opinion, it shouldn't be that explicit, like, yes, you need to do a clinical trial, or no, you don't need to do a clinical trial. Even in the Class 3 PMA universe, a lot of people think that if you're doing a PMA device, you must do a clinical trial. Absolutely. There is no regulation anywhere in the PMA that says you have to do a clinical trial. Obviously, the vast majority of PMA devices undergo clinical trials. And notice I'm parsing my words carefully. I'm saying the vast majority, most of them. I am not saying all of them. It's beyond the scope of this webinar, but there are some PMA devices, albeit not many, but some PMA devices that do not require a clinical trial for regulatory purposes. Once again, remember, average regulatory professionals know the rules. The best ones know the exceptions. So back to the question at hand, does my device need a clinical trial? It depends primarily on the biology and the engineering, not on the regulation. So let's take a more detailed look at this. As I said a moment ago, a clinical trial is never required. If you're working on a 510K device, and the 510K is the workhorse of the medical device industry, obviously here in the United States. The regulation says, this is not guidance, this is the regulation, and this regulation has not changed one word, one punctuation mark since it was created in 1976. Yes, new guidance has come out and people interpret these things differently, but the regulation itself has not changed. So the regulation for a 510K says that a device is substantially equivalent if, in comparison to a predicate, it has the same intended use as the predicate and it has the same technological characteristics of the predicate. So what that means to us in terms of whether or not we need a clinical trial is it has a low regulatory burden. In other words, because it is so similar to the predicate, then the likelihood that we're going to need clinical data to show its substantial equivalence is going to be relatively low. It's not going to be zero. 510Ks do not require clinical data, but notice I'm saying most of them, not all. There are a small but rapidly growing number of 510K devices that do require clinical data for reasons we won't get into here. So the more similar you are to the other device, the less reliant you are on clinical data. And basically what we want to take a look at from FDA's perspective is what the lawyers call the totality of evidence. The totality of the evidence. In other words, all of your evidence, your benchtop testing, your computational testing, if you've done that, your animal testing, if you've done that, and so on, all of that evidence of a clinical trial, if the totality of that evidence is strong enough to justify substantial equivalence, then you're in good shape and you have a very low likelihood of needing to shore up or increase that evidence, the totality of the evidence, by collecting additional clinical data. On the other hand, if your device is not that similar or not as similar to the predicate, you're going to have a regulatory an increase in regulatory burden, and the likelihood or the probability that you are going to have clinical data starts to increase a bit. It's hard in very, very general terms, unless we're talking about a specific device, to be more specific than that. But basically, the simplest way I can wrap this up is, this part of our discussion up, is the more similar you are to another device, the less work you have to do. It's as simple as that. The question is, and I'll leave this as a rhetorical question unless somebody wants to comment on it in the discussion section at the end, is this a good thing? This creates a huge incentives for companies to bring out products that we already have, i.e. a Me Too. And at the same time, it creates a for companies truly new or novel or truly different or hopefully better than what we have before. That's just unfortunately one of the realities of the system. In which a couple of other things to talk. I'm going to wrap this up and move on to the Q&A. How are clinical trials different for SR and NSR devices? Well, there are some basic requirements that were required for all of these studies, and I'm not going to talk about most of them individually. First of all, labeling. You need to have that cautionary statement. Investigational device, yada, yada, yada. Distribution, you can only send it to qualified investigators. What the heck is a qualified investigator? That's up to you to interpret it. Informed consent, monitoring, prohibitions, and records and reports. So these are all components that you need to have right out of the good clinical practices, the GCPs. And I provide a reference on there if you want more on that. How should I select a clinical trial site and an IRB? We talked about this briefly a moment ago. Let me add a little more color to this. It's really the topic of a different webinar, but here's a start. Consider the following: the amount of experience that the investigator or the site in general has with your type of device or similar devices. As a general rule, because the efficacy, and in some cases the safety of a medical device, is a direct function of the skill level of the user, it's sometimes an advantage to go to a site that has a lot of your kind of device or similar devices. Why? Because let me repeat what I just said. The efficacy and sometimes even the safety of a medical device is a strong function of the skill level of the user. In other words, let's consider a catheter. I can put a catheter in one physician's hands and get wonderful results. I can put the same catheter and get abysmal results. The efficacy and even the safety of the device can be is a function or can be a function of the skill level of the user. So looking for sites or investigators that have experience with your kinds of devices is often an advantage. Patient enrollment. Believe it or not, one of the most common reasons why don't finish on time or sometimes don't finish at all is because of lack of patient recruitment or lack of patient enrollment. So make sure that the centers that you choose are going to be able to recruit the number of patients, and ideally with a safety factor beyond that. So if you need 100 patients in your trial, maybe shoot for 150, because you're always going to have what we call lost follow-ups. You're always going to have people drop out of the study for whatever reason. Either they, God forbid, get hit by a bus, or they move away and you have no idea how to contact them. So over-enrollment is common, but make sure that that your sites can recruit those numbers. is consider, as I said before, the average IRB approval time for your particular clinical area, whether it's cardiology or gastroenterology or urology or whatever it is. Usually the shorter the better, obviously. IRBs are required to meet on a regular basis, but the federal regulation, the GCP, doesn't specify how often they need to meet. Specified by the local IRB themselves. Some IRBs meet once a month, some meet less frequently than that. So if you happen to have dumb luck and submit your IRB package the day after that they meet, you might be waiting a month, you might be waiting a few months for a decision. So it's find out when submit your package accordingly. How often do they meet? How responsive or how cooperative are they? Some IRBs are, let's just say, more cooperative than others. It's just a simple reality of life. Are you going to be limited to just traditional IRBs, or can you, pardon me, as I mentioned earlier, consider the possibility of a commercial IRB as well? Consider the economics in academic institutions. These are oftentimes what are called indirect costs. And sometimes the indirect cost minimum can be about 40%, and they can go up. I've seen, as high as like 80 to 85%. So basically what that means is if you're doing this at an academic center at a university, for example, just take the in terms of actual costs and basically double it, because that's the amount of money that you're going to be paying the university, and they're going to take that indirect cost and use it for overhead and who the heck knows whatever else. And another factor to consider is the PR value of the investigator and or the site that you're using, and are there some key opinion leaders at that particular that will help you with the clinical adoption problem. In other words, getting your device through the FDA and onto the market, in many ways, that's the easy part. Getting physicians to use it, that sometimes is a challenge. And let's not be so naive as to think that just because FDA approves a product, physicians will necessarily use it. On the contrary, I can give you lots of examples where have gotten through the FDA and onto the market, but for one of a variety of reasons, most physicians don't use it. So these are just some of the parameters to think of. Obviously, there are other things as well, depending on your situation, but some of the things to consider when you're selecting your clinical trial site and your IRBs. Should you do a pre-submission meeting or a pre-FDA for an NSR device, even though it's not required? My short answer is almost always yes. The vast majority of times, I recommend doing a pre-sub. If it's an NSR device, even though it's not required to do. Remember, NSR devices are IDE exempt. And why do I make this? Because as I said before, I want to mitigate or possibly eliminate two common problems I see throughout the industry. One is I want to confirm that FDA agrees on my NSR determination. In other words, I want to avoid that scenario that I shared earlier where you determine your device is NSR, the IRB agrees that it's NSR. They say, gee, Mike, I don't know what the heck you're smoking. You must be smoking your socks or whatever it is that you all smoke in California, because we don't see this as NSR. Right. So I want to avoid that scenario. It doesn't happen often, but it does happen. The other scenario I want to avoid, which is much more common, is I want to confirm with FDA on my clinical trial number of patients, number of sites, inclusion and exclusion criteria, and most especially the clinical endpoints that we measure and the clinical endpoints that we don't measure. Because I don't want to be in a situation where I measure certain clinical endpoints and I submit them to the FDA, and FDA says, gee, we'd like to see these one or two other endpoints. And as I said earlier, clinical trial all over again to collect the information on those additional endpoints. Can you say ka-ching, ka-ching, ka-ching? As I said, and I don't mean to be harsh, but if we're going to call ourselves medical device professionals or regulatory professionals, that word professional should mean something. Just like a surgeon is a professional and he or she is held responsible for not just medical device professionals, and regulatory professionals should also be held responsible for what they do and what they don't do. And to make that kind of an amateur or elementary school, or what I like to say boneheaded mistake, is in my opinion, it's a hard thing to understand. So to wrap this up, just a couple more and Q&A. So please be typing in Q&A questions into the chat, and Eddie, and you can triage the Q&A questions and pick out the ones that we'll have time to discuss. What are the challenges for the future when it comes to SR versus NSR determination? Well, just one, can you say personalized medicine? And medicine in the medical device world, not in the drug world, but in the medical device world, is three-dimensional printing, or 3D or 3DP. Here's an example of a stent. Can we print a stent? Well, first of all, what kind of stent are we talking about? A bare metal stent? That's boring. A stent graft? That's pretty boring too. How about a drug-eluting stent? Well, that's a little more kind of boring. What if we can print a customized, or in other words, a personalized bioabsorbable stent? In other words, we don't take the stent, from the manufacturer out of the package and put it into the patient. Imagine this not so hypothetical scenario. We're doing say a cardiac catheterization in the CCU, in the cardiac cath lab. Determine the patient needs a stent, or maybe multiple stents. We further determine that we want to have a drug-eluting stent. And we have in the hospital, or even in the same room as we're doing the catheterization procedure, we have a 3D printer that will print a customized stent. In other words, a stent that is designed to put in that only in that particular patient. In other words, it has the size, the diameter, the tapers, and so on. It has the combination of drugs. Why are we limiting drug-eluting stents to only one drug? It makes absolutely no sense. Even though we've been doing it now for a decade, it makes no sense to me. Put multiple drugs, and not just drugs, but biologics on that particular stent, which, by the way, it's not a medical device. And if that's not cool enough, we do all of those things and we make it out of a bioabsorbable material, so that once it delivers all of the drugs, not only does the drug go away, the stent goes away. Think this is Star Trek-y? Absolutely not. I have a couple of customers that are working on exactly this kind of technology. Just as a quick example, watch this. And I'm going to, for the sake of time, skip the first couple of seconds. Here you see a 3D printer, and at the bottom, I hope you're seeing the video here, okay. At the bottom, you're going to see a stent coming out of the 3D printer. And this could contain the drugs. It could be bioabsorbable. It can be lots of different things. Let me just jump ahead a little further. So there you can see the stent there. So imagine we're doing this right in the cath lab. We take the stent out of the 3D printer. Maybe we rinse it off or do a little, whatever we want, and then we stick it right into the patient and we're done. This whole concept of making devices in a manufacturing facility somewhere and then putting them in a package and shipping them, to some other place in the world, even though we've been doing that for a very, very long time, and that is not the future of medicine. When it comes to the SR versus NSR determination, well, for most devices, if the device SR versus NSR determination has been made. In other words, I mentioned earlier in one of the examples that intravascular stents, like a coronary stents, are deemed significant risk devices, whereas urethral stents, for example, would be an NSR device. But assuming that we're printing a coronary stent, an intravascular stent, then that's going to be a significant risk device, most likely. Because all we're doing is we're changing the manufacturing method. We're not changing the indication or anything like that. However, interesting question that, as far as I know, nobody has crossed this bridge yet. Could changing the manufacturing method flip the SR-NSR determination? In other words, let's say that we have a device that is an NSR device that is but now we change the manufacturing method such that it's 3D printed as opposed to injection molded or whatever it was before. Could that potentially flip the designation from NSR to SR? Absolutely, yes. It's certainly possible. Remember, the SR and NSR criteria go back to at least 2006, probably before that. And clearly, 3D printing personalized medicine technologies didn't exist in those days, so the regulation could not possibly address this. There's nothing in those 4 requirements of the SR determination that I told you from the CFR the other day that, or earlier in the webinar, whether that even remotely hint at the manufacturing method. We're going to have to add into the CFR. I suspect probably yes, because sooner or later this is going to become a problem. But in my opinion, of course, this has not happened yet, but there is definitely the possibility that changing your manufacturing method, in this case to 3D printing, could flip your SR and SR determination. And when that happens, whether it's a few months from now or a few years from now or whenever it is, just remember you heard it here in this webinar first. The last thing that I want to mention, and then we're going to wrap this up, because we're going to have the last remaining minutes for Q&A and discussion. Is there a template for the SR versus NSR determination? Well, the answer is yes. If you Google significant risk, for example, you're going to get a laundry list of different things. The question is, would you, I'm sorry, it's supposed to say would you use, it's not would you sue. Sorry, that's a typo. Would you use any of these? Well, I can't speak for anybody else, but I certainly would not, for many reasons. But I know nowadays a lot of opinion, especially as an engineer, I loathe the idea of templates. I don't like FDA giving us templates because it limits significantly the freedom that I have to present the information that I want to present in the way that I want to present it. And maybe this is the former R&D engineer in me. If my company asked me to design a particular, I'm going to turn around and ask them, well, can you please give me a template that I can fill in to design my device? If you ask them for that, they're probably going to fire you on the spot. So you know, using templates, you know, is the quintessential example of thinking inside the box, of, you know, not having the ability to express creativity or to have imagination. Over the last many years, I've developed a series of my own templates that I share with my customers. This is not part of the handout because I don't typically give my templates away pro bono, but I'm just going to let you take a look at this real quick. It should be a standalone document. In other words, somebody that's not familiar with your product should be able to read the document and understand it. You should have an overview and introduction. You know, what is the purpose of this? Especially if it's an NSR, this is an NSR justification, so there's no formal review of the device by the agency. You need to really make sure that you have in your device description section the technical details, biocompatibility, electrical safety, sometimes the only thing that for an NSR device, the only thing that stands between the device and the patient is the IDE. The FDA is not there. You can include a brief section on your regulatory strategy. You don't have to, but I usually do. 510K, de novo, PMA, whatever it is. Most importantly, you need to have your labeling. Earlier, labeling a device with one indication might be an NSR. Labeling the same device with another might be an SR. And of course, the most important part is the actual NSR determination. These are the requirements from the CFR that I showed you earlier. So this is the same way as I do the 510K flow chart. I take them one at a time. The device is intended as an implant. You provide a response. Yes, it is an implant, or no, it's not, and here are the reasons why. Represented to be supporting or sustaining life. Yes, it is, or no, it's not, and here's why. By the way, these are not yes or no questions. You have to be able to explain your answer. It's kind of like in elementary school, you know, in math class, show your work. 1 + 1. How did you get to the answer 2? Obviously, that's a fairly trivial example, but I assume everybody knows what I mean. And similarly for the others. And then I also include in here a section on risk and how we mitigate those risks. And then finally, a summary and conclusion section. And then I provide additional resources from FDA and from me and so on. So this is my NSR justification template. You certainly can use a different format if you want to, but I can tell you that I have had a lot of success using this. If you want an actual copy of it, talk to me about becoming a customer. So if you want even more, then if you ever seen the haven't, let's just lighten things up a little bit with a few seconds of this video, and we'll the Q&A. Oops, sorry, gotta back up to the beginning. Here we go.
I don't think we're hearing the sound.
Oh, we're not? Oh, I'm sorry. I tested it earlier. Oh well.
No,
All right. It's not that important.
we'll put the link in the if you have the link in the slides, people will be able to check that out later. That's
okay. Cool. Thank you for letting me know, Eddie. Anyway, so if you want more, I've put out a lot of resources on risk, significant versus non-significant risk in particular, with Greenlight as well as other organizations. So feel free to take a look at those. And if you have any questions, you can certainly contact me. And last and certainly not least, this is the guidance that FDA has put out on this particular subject. Notice I put the word current in air quotes, because if you look at the bottom here, that's the public date for this guidance is 2006. That is, I confirmed this yesterday, that is still the current guidance. So I guess it depends on how you define the word current. Have you had enough? So just to wrap this up, what are the takeaway messages in no particular order? Ones. Remember that your SR and NSR determination is not up to the FDA. It's not up to the IRB, at least not directly. It's up to you. You need to make that determination. And then once you do, you need to go to the IRB, and in some cases, go to the FDA and sell it to them. Remember that an ID is required for SR devices, but for for NSR devices, an IDE is not required. You're IDE exempt. But as I've talked about now a couple of times, even for an NSR device, I almost always suggest that the company go to the FDA clinical trial, even when it's not required, to avoid those two common problems that we talked about earlier. Also, begin your investigator site and your IRB selection process early. I can just about guarantee that it's going to take you longer than you think. In spite of using my equation for including that safety factor, that safety margin, that fudge factor, if you will, it may even take you longer than that. On the other hand, if you get your sites and your IRBs identified in advance, and it's a couple of months before you begin your clinical trial, then no problem. Nobody's going to worry about that. But if you're ready to begin your clinical, the IRBs have not bought off on that yet, or you haven't even begun to line up or qualify your clinical investigation sites yet, that's a problem. And suffice it to say, I don't think the senior management in your organization is going to be too happy about that. And of course, there are a lot of other things that we talked about in the webinar, that may or may not be important to you depending on your particular situation. So we've covered. Just want to wrap up with one of my favorite quotes from Hillary Clinton, not to get into politics, but obviously we're in a political season now. She said, "There's nothing magic about regulation. Too much is bad. Too little is bad. What's the right amount of regulation?" Well, here's one of my several regulatory sense. Then we shouldn't need it. If the regulation doesn't make sense, we shouldn't have it. It's as simple as that. And of course, I came up with this, so I'm biased, but I think this is absolutely brilliant. If the regulation makes sense, we shouldn't need it. It should be a statement of the obvious. If the regulation doesn't make sense, then we shouldn't have it. In other words, why do we want to do things that don't make sense? Remember, I used to teach medical school back in the day to say this to my med students a lot. The surgery went perfectly, but the patient died anyway. Well, the regulatory equivalent of it is we followed the regulation perfectly. That is, we did all that FDA or Health Canada or whoever asked us to do, and yet the patient died anyway. Unfortunately, the solution to this problem, in my opinion, is not to create more regulation, because we got thousands and thousands. Is to get people to think. Present company excluded. So what's the message here? Do what makes sense. Whether it's required or not, it doesn't matter. Do what makes sense. Or maybe I'm just naive. Maybe I'm just living in the universe of rainbows and unicorns. But I definitely think this is possible. So with that, I want to just skip these last couple of slides. I want to thank everybody for attending this webinar. I'm happy to stick around for the remaining time that we have to answer questions and have some discussion. But for those of you that are not able to stick around, or for those of you that for whatever reason don't want to ask your question during the webinar, you have my contact information, phone, email, and so on. Feel free to help however I can. I want to thank everybody in the audience for attending the webinar. I want to thank my many friends at Greenlight Guru for giving me the opportunity to conduct this webinar. And with that, Eddie, and I'll turn it back up to you to facilitate the Q&A discussion.
Sure thing. Thank you, Mike. I know you covered a ton of ground, so thank you so much for the fantastic presentation. There are a lot of questions. We're obviously not going to be able to get to all of them, so I will just let you all know, those of you who've asked questions or voted on questions, we'll provide those to Mike so that as he's able to, maybe he can get to some of those and respond to them one-on-one. There has been a request for all of the Q&A to be provided. I don't know if we'll be able to do that, but I'll show that to our team. So, it's an interesting request I don't know that we've gotten in the past. All right. One of the questions that seemed to be kind of good for a clarification question was from Victoria. I think you've answered this, but does the device have to meet all the requirements of SR, or just one to be SR? And I'll share it with you so you can read it yourself.
Yeah, good questions. So what I address each of the four requirements. Clearly, there's going to be situations where it does not meet all of them, but I believe I won't take the time to go back to actually look at what the CFR says. But if you parse those words carefully, does it have? Well, let me see if I can.
Sure.
I should be able to. I just want to check the exact verbiage from the. Okay, so here it is. It's on my slide 14, but it might be different in your handout. SR means an investigational device that is that, or otherwise that. So actually, it's a good question, Victoria. I would go back. I would actually take this a step further. If I click on this hyperlink, if it works, it takes me right to this particular section of the CFR. And I want to remind everybody, this is the CFR. This is not guidance. So I always go to the CFR first, because the CFR is binding, the guidance is not. Let me just skip down to, here, subpart M, significant risk device means an investigational device that, and then it lists these four things. So you know what? And this is interesting, it does not explicitly say it must meet one of the criteria or all of the criteria, but I think, Victoria, in my opinion, it's not an all or nothing thing, because it certainly could be that your device is an implant, but not one of these other things. So I would say that as long as it meets one of these criteria, then it would be significant risk device. But once again, of words and your ability to defend your interpretation. In this particular case, there are no words, so you have to be able to interpret what the regulation is not saying here, and then defend, your interpretation of what the regulation is not saying. I'm not sure, Victoria, if you would agree that that's a good answer to your question, but it's the best answer I can give. But it was a very good question. Thank you for asking.
Yeah, that was a great question. It's also a good answer in that you showed where she can go to dig more. So I think that's great. Verna asked a question that, again, maybe something that could stand to be clarified just because it was upvoted. quite a bit. I just noticed the NSR NR is only used in describing investigational devices, if that's correct. So is this what distinguishes the categorization for 14971? So I thought you might want to clarify on that.
So that's a good question. So in this context, because the word investigational has many different connotations, but in this context, investigational normally means that the device is not yet on the market. However, remember, as I've said a couple of times now, average regulatory professionals know the rules. The best ones know the exceptions. Let's say, hypothetically speaking, and I've been involved in several of these quote unquote hypotheticals, you have a device on the market for one particular indication that is NSR, and now you want to do what we call a label expansion. You want to do you want to add another indication to the label, and that second indication is a significant risk. It's not a non-significant risk, even though the device itself is the same. That is what it does, how it works, it's a mechanism of action. Because now we have a different indication, one that is NSR and another that's potentially SR. Now you might have to do an IDE. As a matter of fact, let me strike the word might. You will have to do an IDE because in the eyes of the law, even though the device is the same, because the label is different, it's a new device. I know it doesn't sound to a normal person, well, it's the same device, it does the same thing or whatever, but because the label is different, it's a new device. So it could very well be that you have a situation where one version of your device is IDE exempt, is NSR, and another version of exactly the same device with a different indication is not IDE exempt because it is SR. I hope that answers the question. Edian, if you want to add something, feel free.
Yeah, no, it's good. I know there are a lot more questions, and we're not going to be able to get to any more really. So I really appreciate you answering those. But we'll provide those to you. So if you're able to get any additional clarification to the additional Q&A, then we can do that. But
Edian, is that a kind reminder that I'm being a little long-winded here?
Not that you're long-winded. I think people would appreciate the insights. So the Q&As have really helpful for very specifics, but we are getting close, and I always like to give people at least just a few minutes between meetings. So thank you, Mike, for the very informative presentation, all the valuable insights that you provided. As a reminder, because we do always get this question throughout and even after, we will be sending you today's recording, those of you who are listening, and the presentation slides. So you'll be able to check those links out and do a little bit more digging. On behalf of Greenlight Guru, I want to thank you all for attending today's presentation. And yeah, Mike, thank you so much. Always enjoy having you.
Thank you, Edian. I appreciate it.
All right, everybody, take care. We'll let you get back to it.
Bye-bye.